All right, so last week we saw what kind of neurons there are. Now we need to know a little bit more about their structure. It'll actually take us several days to talk about what a neuron is made of and how a neuron works, because there's a whole bunch of things that go into this. Today we're going to focus on structure, so what are the components. Then we'll move on to talking about how nerve impulses are generated and how they travel, and then how they get from one neuron to the next neuron. So there's a whole bunch of detail that goes into that. Now, if we talk about the structure of a neuron, there are some pieces that are common to all neurons. So I don't use the word all lightly. All neurons are supposed to have these parts. Of course, someone could have a disorder or someone could have damage that could destroy one of these things, but neurons are all supposed to have these pieces. Then I'm going to add to this list with some components that only some neurons have. Now, the first thing on the list is dendrites. And dendrites is this part here. They look like tree branches extending off the main part of the neuron. So if I want to know what a dendrite is, dendrites are short, branching terminals, just like they appear on the picture. It is very probable that at some point you would have to identify on a picture which part of it is a dendrite. And so you want to look for those tree branch resembling structures. They receive is important, and relay impulses. Now, if you look at the top of that picture where the dendrites are and at the bottom, they both kind of look like branches. But if you think about a neuron like it was a tree, these are like the branches and the part at the bottom looks kind of more like the roots. Another way you can recognize which things are dendrites is by recognizing that they surround something. So I like to mention that they surround the next thing that we'll talk about. So if you're ever unsure, I see some short branching things, are they the dendrites or are they something else? You're going to look to see, well, are they around the cell body? Because if they are, then they're the dendrites. So we're just looking at one neuron in a picture here, but if there were more, the end of the other neuron would be attached to the dendrites of this neuron. So when I say that it receives and relays impulses, that means another neuron, some other structure, would be sending an impulse to it, it would receive it, and then it would send that information along in its own structure. Now, of course, I just added one other neuron, but there could be 500 other neurons that are attached to it. So this is not just a linear pathway. Each neuron could have hundreds of other neurons sending impulses to its dendrites. The second thing on the list is the cell body, or the soma. Anytime you see S-O-M in a word, some, like a somatic cell, it means the word body. And that is this part here in the middle. You'll usually recognize it because that is where the nucleus is located, and the nucleus is generally represented by a dark circular structure. So in the cell body, that is where the nucleus is. That is also where any sort of metabolism that's going to happen in the neuron occurs. So neurons, like any other cell, need to stay alive. And so in the cell body, that is where reactions that would, say, break substances down, produce wastes, generate energy, all of that stuff would happen in the cell body. The other thing that happens is that it continues to relay impulses. So the dendrites got an impulse, a nerve impulse, from somewhere, and they send it to the cell body. The cell body relays impulses after adding. What that means is, it's going to consider information from all of the other neurons. It's sort of like the command center. The neuron might get a bunch of different signals from a bunch of different neurons through its dendrites. And the cell body's job is to sort of add them all up and see, in total, is there enough here for me to continue sending an impulse? Or was there one telling me to send one and another equal one telling me not to send one, and they cancel each other out? So that's what I mean after adding. It's got to take information from all of its dendrites and see, is there enough here for me to continue sending the impulse? The third piece is the axon. Now, the axon is this skinny stem here that runs from the cell body down toward the end. There's some stuff on the axon because it's really hard to find a picture of a neuron without it, but not all neurons have the same covering over top of their axon. Now, if we think about what the axon does, what its job is, the axon conducts the impulse. So if the cell body, after receiving impulses from all of the dendrites, decided, yep, it's time to send an impulse on, it's going to travel the length of the axon. Now, the axon can be long. It can be up to, and you might be surprised, but it could be up to 1 meter long. Most of them aren't. Most of your neuron axons are in the millimeters or micrometers in terms of length, but there are some really long ones. For example, your sciatic nerve has a really, really, really long axon. It goes from up here at the base of your skull all the way down. So 1 meter would be the longest, but most of them aren't that long. The fourth thing that's common to all neurons is the piece that's at the bottom. So this whole section here, that if we're continuing with the tree analogy, looks like the roots of the tree. Those are the axon terminals. Sometimes they just get referred to as the terminals. And sometimes, instead of calling them axon terminals, we'll refer to something that isn't really a structure. It's called a synapse, not to be confused with synapsis, which was the name of the process where homologous chromosomes lined up in prophase 1 in meiosis to make a tetrad. Not the same word. Sounds exactly the same. The only thing that's different is the E, right? It's an I when we're talking about chromosomes, and it's an E here. The axon terminal, like its name suggests, is at the end of the axon. So it has branches just like the dendrites. And it releases a substance for communication. Now, where does it release that substance? Into this mysterious synapse. And the synapse is the space between two neurons. So the synapse is not a part of the neuron. It is the space between one neuron and the next. But like I said, sometimes when we're referring to axon terminals, we'll say something about the synapse to give you the idea that that is the end of the axon that we are at. Now the other thing that's sort of vague sounding here is this substance business. If I consider the first 3 parts up here, I called it an impulse. That's implying that it has something to do with electricity. It's an impulse. When we get to the axon terminals, now we're talking about a substance, a physical thing, not a signal, not an impulse, but rather a molecule that you could identify that is going to travel from the axon terminal, from those little branches, from the neuron that you have to the next neuron that's in the pathway. Now, those are the 4 pieces that are common to all neurons. Does anyone want to ask anything about any of those 4 pieces? Okay. So aside from those four, there are some other structures that only some neurons have. So you will not always find these structures. The first of these is the myelin sheath. Now the myelin sheath, we'll call this number 5, is this thing that looks like it's wrapping itself around. Now the myelin sheath is this layer of tissue, and the way it looks, I feel like it kind of looks like I'm wrapping it up, like keeping it warm. And it's an insulator, but not for temperature, but rather for electricity. So the myelin sheath's job is to be a fatty, insulating layer and it goes around the axon. Now I'm sure that some of you have maybe seen a piece of meat with some fat on it still. Does anyone know what color the fat is? Yeah, it's white. So I'm just going to point this out here. Even though the cell in my picture looks kind of green, the myelin sheath makes the axon look kind of white, just like fat looks white in a piece of meat. Now, where does this myelin sheath come from? Well, it comes from a cell. So this thing up here, this cell, so I can see a cell body there, and it looks like little pieces of it are extending outwards and becoming the myelin. Those are Schwann cells. Schwann cells are glial cells. So we saw those last week. Glial cells are the support cells. Remember, glial comes from a word that means glue. So they're providing support. And very specifically here, they are forming the myelin sheath. So those glial cells are specifically providing this layer of insulation. Now, if the entire axon were insulated, it would be really difficult to get the electrical signal to pass, right? This is insulating the axon. So there need to be spaces. So this next thing on the list, number 7 there, is a space on the axon between one piece of myelin and the next. It's called a node of Ranvier, or Ranvier if you're going to actually say it properly. I don't care how you pronounce it. So these are the spaces between the myelin sheath. And then the last one, we'll call it 8, is something called neurolemma. Now, you might have heard a word in Bio30 called sarcolemma. Anyone have an idea what neurolemma might mean or remember that word from Bio30? It was about muscles. There was the sarcolemma, the sarcoplasmic reticulum, the sarcoplasm, all those words that sounded the same. Does anyone remember what it is? Well, that's where it's coming from. That's where the word sarcolemma was coming from. So it was a structure that was part of one muscle fiber, so one muscle cell. Anyone remember what piece? Yeah? That was the sarcoplasm, but you're getting closer. It's an organelle that's in a cell. It's not the cytoplasm. Someone just whispered it. It's the membrane. So neurolemma is a thin membrane on myelinated axons. So if you look at that myelin, there's this very thin outer layer here. That's the neurolemma. Neurolemma isn't always identified in pictures because it's realistically part of what makes up the myelin. But here's why it's important. A lot of researchers have been looking into whether the presence of neurolemma makes it possible for axons that were damaged to regenerate after injury, which is a big deal. Initially, people thought if you damage a nerve, it is never growing back, ever, ever. But what it seems though like is that neurons that have this neural lemma on it may be able to regenerate. Now, there is a lot of research, a lot about nervous system regeneration. There are a lot of people who spend their lives figuring out ways for people who have damaged or completely severed nerves or neurons to regain function in the part that was damaged. So probably the most severe form of this you would have heard is spinal cord injury, where if your spinal cord is damaged, you are paralyzed, and that is it, end of story, it's over. But maybe not. Maybe not, because some nervous cells seem to be able to grow back or regenerate even after damage has happened. And this neurolemma seems to play a role in that. If you haven't thought of your de facto topic yet, you could make 18 billion de factos about your nervous system. I would be super duper interested in every single one of them, because remember, this is my jam. So folks, we're going to fill in this little chart that helps us compare a myelinated versus an unmyelinated neuron, because remember, not all neurons have the myelin sheath surrounding the axon. Now, I told you earlier that myelinated neurons make it look white. So if we're talking about white, or sometimes you'll see the term white matter, we are talking about myelinated neurons. Unmyelinated neurons are referred to as gray or gray matter. Now, if we ask ourselves where we might find myelinated and unmyelinated neurons, in general, in your PNS, all of the nerves should be myelinated. Yep. This is the word all. Okay. They should. That doesn't mean that they are, because like I said, there are disorders that could cause the myelin to become destroyed. For example, multiple sclerosis is one of those, where the myelin sheath becomes damaged or destroyed, and it affects the way your nerve impulses travel. In the central nervous system, there are also myelinated neurons, but it's not all of them. It is the outer spinal cord, but the inner brain. So you've seen a picture of a cross-section of a spinal cord, and it looked like there were two different parts to it. The part that looks like an H and the part that surrounds it. The part that looks like an H is the darker part, and the part around it that looks whiter is white matter. If we look at unmyelinated neurons, they should be found generally in your CNS, not in the PNS. So central nervous system, not peripheral nervous system. And they are found in the inner spinal cord and outer brain, which we'll see later is called the cortex. Cortex means bark, and lots of things have a cortex. So if I said that I had an unmyelinated neuron, the number one choice for where it is located is your central nervous system. It should be there. The only reason we would say that one is in the PNS is if damage had happened, but if we're looking for gray matter, we should find it in the CNS. White matter is found in both the CNS and the PNS. Myelinated neurons make for faster impulses. Now, it might seem weird that something that's insulated works faster, but because only small bits of the axon are exposed, those are the parts where the impulse travels and it jumps between them. Unmyelinated neurons have slower impulses. Now, if we continue on that vein of impulse traveling, the faster impulses are because of something called saltatory conduction. And the word saltatory means jump. So when the nerve impulse is traveling down the length of the axon, on a myelinated neuron, it can jump from one node to the next. It can skip over the parts that have the myelin sheath. Unmyelinated neurons have non-saltatory conduction. And even though we put it on the picture, I always like to add here, the myelinated neurons have neurolemma, and the unmyelinated ones don't.